We are going to conduct the restriction digestions of the chromosomal DNA and the plasmid DNA that we isolated in the previous experiments. We will analyze that what happens when we cut a plasmid DNA with the restriction enzymes and what happens when we cut a high molecular weight DNA like chromosomal DNA with the help of restriction enzymes. So our experiment is restriction analysis of plasmid and genomic DNA. So this is the experiment that we have planned, and our template DNA, genomic DNA, is the weak DNA that we isolated in experiment number 5. And this is the plasmid DNA that we isolated in experiment number 3. So what the restriction enzyme that we will be using in this case is PVU2. This enzyme is a hexanucleotide cutter and will cut CAG, CTG. And will generate the 5' overhangs. We are not going to look into the 5' overhangs, but we will see that if we digest this plasmid, which has got 2 sites on it, okay, this plasmid that we are going to use, that we isolated in experiment 3, is PSD Dna2. This one is the plasmid, okay? And this has got 2 sites for PVU2. So what happens if you cut a circular DNA at 2 points? You get 2 fragments coming out. We are expecting 2 fragments in this case coming out of the plasmid DNA because we know that there are 2 sites on them. One of the fragment is actually 1.5 kb and the other one is about 10 kb in size. So you will see on the gel a 1.5 kb fragment and a 10 kb fragment, which will be coming out of this. On the other hand, what will happen when we cut the genomic DNA using this PVU2 enzyme? Should the genomic DNA give us discrete sets of fragments as we get in plasmid DNA? Okay. So what I mean is, if we cut the genomic DNA, with PVU2, what should we expect? Discrete fragments or some other kind of pattern? And if some other kind of pattern, then why that pattern should appear like that? Okay. So what happens is that if we cut any kind of genomic DNA with a restriction enzyme, whether we cut it partially or completely. Partially means for a couple of hours, and complete digestion means and overnight digestion. Okay, so if we cut and load that DNA into the well of an agarose gel, we see a smeared pattern. There is never a discrete DNA fragment that can be seen due to the digestion with restriction enzyme, particularly in case of genomic DNA. And the simplest reason for that is that PVU2 does not exist 1, 2, 3, or 4 times. It exists a 6 nucleotide cutter. So every 10 to raise power 6 nucleotides, there will be a restriction site for this enzyme on the big genomic DNA. And that generates actually nested sets of fragments which are very close in sizes, and they cannot be actually separated as single fragments on the gel. Okay, so they make a smeared pattern always. That kind of digestion always makes a smeared pattern because of the thousands and thousands of restriction sites of this enzyme on the whole genomic DNA. And that's what we are going to realize today. Now, regarding the restriction setup, what we will do is that last time we saw that our chromosomal DNA was close to the well after running about 30 minutes. On the other hand, if we remember the experiment number 3, the plasmid DNA appeared as a couple of bands, and it migrated very well on the gel because it was a small molecule. Okay, so the restriction setup, we know almost the concentration of the plasmid and the chromosomal DNA, and we loaded about 1 to 2 microliter of our preparation on the wells for that purpose. So in order to see that very clearly, we will use plasmid, we will use about 3 microliter of the plasmid today. The restriction enzyme, which in this case is PVU2, okay, we will use 1 microliter of that, and then we will have the restriction enzyme buffer. These buffers are very specific to the restriction enzyme. Okay, they are low salt, medium salt, or high salt buffers. Okay, so we will select the buffer according to the restriction enzyme. So they already know what is the concentration they have already prepared. So this is always supplied along with the restriction enzyme, so that we should use that buffer when we are performing the restriction digestion. So in a 10 microliter reaction, these buffers are usually 10x. In a 10 microliter reaction, we use 1 microliter of the buffer. But today we will be performing a 20 microliter reaction. Okay, so 10x buffer for 20 microliter, it will go like 2 microliter per 20 microliter. So if we calculate them all, the total is 6. We have used 6 microliter of the components, and then the fourth component in a restriction digestion reaction is double distilled autoclaved or sterile water. So we will use about 14 microliter of that, and the total will become 20 microliter. The same will be the case with genomic DNA, but instead, in case of genomic DNA, here we will use genomic DNA, and that will be 5 microliter per reaction. Restriction enzyme will remain the same. Buffer will remain the same because our total volume is going to be 20 microliters. Okay, so we increase this genomic DNA by 2 microliters compared to 3 microliter in plasmid. So we have to decrease 2 microliter from water. So we will add 12 microliter of water, and this will total into a 20 microliter reaction. This reaction will be incubated. It can be incubated overnight. It can be incubated for about an hour. But we will do a partial digestion of the genomic DNA for about 30 minutes. And same thing for the plasmid DNA. Okay, 30-minute digestion of plasmid, 30-minute digestion of genomic DNA. I'm quite sure that the plasmid DNA can be digested very well in 15 minutes because 1 unit of a restriction enzymes digest 1 microgram of DNA in 1 hour at 37 degrees centigrade. So we are not taking 1 microgram of DNA. In our case, the DNA is around 0.25 microgram. So it will be cut in 30 minutes very quickly, very fast. On the other hand, the genomic DNA cannot get cut to completion in 30 minutes. We need to incubate for overnight for getting the complete digestion of the genomic DNA. So 30-minute digestion will result into partial digestion, and that is important to see rather than going for complete digestion. Okay, so the reason is that the partial digestion means all the genomic DNA is not completely cut by PVU2, and we can see a lot of genomic DNA close to the well, and then the remaining DNA which has got cut by the enzyme. This is the thick band of the chromosomal DNA that I'm expecting to see in the well after partial digestion, and then some smear appearing from here. That smear is not that long. If we incubate it for overnight, okay, then what happens is the smear gets too big, indicating a complete digestion. Okay, so you can see the digestion here. You can see the digestion here for 30 minutes. I'm expecting like this. Okay, so now we will be going to the lab and we will see how we perform these reactions. And when the incubation period is going on, we can pour a gel, and then by the time the incubation is over, we can run the gel there. Okay, see you in the lab then.
We are set for proceeding with the restriction digestion. What we have got here is the PVU2 enzyme, and we have its buffer, which is PVU2 buffer G. Okay, and this buffer will be used, which is supplied by the manufacturer. And if you notice, the PVU2 enzyme, it comes in a tube which is green tagged on the top. And one of the way of identification of the right buffer for a restriction enzyme is that the buffer will also have the same green cap, so that you can easily pick that from the freezer without disturbing the temperature for the other restriction enzyme that you are going to use. So these enzyme and buffer, they are placed on ice. We are having double distilled water, and we have the chromosomal DNA with us, which should also be there on the ice. And then we have the plasmid DNA. We have the plasmid DNA here in these tubes. The FSD8 is actually the wheat variety, which we used last time for the isolation of the chromosomal DNA. And in this tube, we have ultra pure water. Okay, so we will use this water to complete the 20 microliter restriction reaction. And what else we have got is we have 1 kb DNA ladder, which is here in the rack. You can keep it at room temperature, or you can keep this also in ice. So I will prefer to take it to ice. These are the things. Then we have the gel loading die, which we will use actually when we are ready to load our samples onto the gel after restriction digestion. Okay, then we have some micro tips, which are 20 microliter, and then we have a 200 microliter tips rack, a band of tubes for setting up the reaction. They are there. We have the pipette pens of small volumes, because restriction digestion usually requires small volumes. And we have a 10 microliter pipette pen and a 100 microliter pipette pen here. We have the gel electrophoresis, where we will pour the gel, and after digestion we will resolve it, and then we need a power supply to run our gel. We have got here is ultra pure agarose, and then the flask in which we will prepare the gel, and this is the cylinder for measuring 1x TAE. So we are now ready to proceed for the experiment. Now she is labeling the tube before we set up the restriction digestion. So we will be performing restriction digestions in 2 tubes. In one of the tube we will carry the plasmid for restriction digestion, and in the second one we will take the chromosomal DNA, that is the total genomic DNA from wheat. So that she can label it like plasmid and genomic DNA, or she can simply label it like tube 1 and 2, but keep the record on. But what she has done is that she has labeled it as plasmid DNA, and the other tube she has labeled genomic DNA, that is gDNA. Okay, so both of these tubes will go to the ice, where she will set up the restriction reaction. Okay, she is ready to do the restriction setup, and she has taken one of the tube that is for the plasmid DNA. And if you remember, on the whiteboard I wrote about the constituents of the restriction digestion mixture. So she is taking the plasmid in one of the tubes, so the water is about 14 microliter, the plasmid is 3 microliter, the buffer is 2 microliter, and the restriction enzyme is going to be 1 microliter. Okay, so she will be pipping out all these constituents into the same append-of tube, which she has labeled for plasmid. Okay, so one by one all these ingredients are going to be mixed. So now she's going to take the buffer, that is buffer G, particularly developed for PVU2 restriction enzyme, and she's mixing that buffer with the other constituents. So far she has added the plasmid, water, and the buffer, and at the end she is going to add 1 microliter of the restriction enzyme to this tube, and that will be done. That's PBO2, 1 microliter, and this is being added. So all the 4 ingredients are complete now in the plasmid tube. So that tube is ready for incubation at 37 degrees centigrade. But by the time she will put it in there, she will set up the second reaction so that we can transfer both of the tubes together to 37 degrees centigrade water bath or a hot plate. She's just trying to mix the materials well, and now she's performing the restriction digestion of the genomic DNA. So she has taken 5 microliter of the genomic DNA into the tube, and now she will add 12 microliter of double distilled sterile water to the genomic DNA so that it is ready for the buffer and the restriction enzyme. Okay, both the genomic DNA and water have been added to the tube. So she is taking now the buffer from that green cap tube and adding it to the digestion mix for genomic DNA. The buffer has been added. And the last step is to add restriction enzyme. So this is just a 4-step procedure. It's not complicated at all. So the only thing is you have to have the genomic DNA pre-isolated or the plasmid DNA pre-isolated, and then only the 4 ingredients are added to the restriction digestion mix. Now she has taken the restriction enzyme and adding it to the tube so that all the 4 ingredients are now complete there. She's just mixing that, okay? And after mixing it, she can place this tube at 37 degrees centigrade. Remember that all the molecular biology enzymes, they have got 50% glycerol in it. So when you put the enzyme in a tube, it settles down at the bottom. So it's very essential that we mix the ingredients so that the restriction enzyme gets suspended evenly and we can then proceed for the incubation time. There's a 37 degrees centigrade a hot air incubator. What she's going to do is that she is going to put these tubes now in this incubator, which is preset to 37 degrees centigrade, and we will incubate these tubes there for about 30 minutes, and then we will come back and see the digestion on gel. The gel has been poured and it's almost solidified. We are ready for loading the samples of the restriction digestion into this gel tray. Everything's almost set. We have the gel loading buffer here that will be loaded into the gel chambers. The apparatus will be loaded with this buffer shortly. As soon as we we get our restriction digestions out of the incubator. The power supply will be set to 60 volts for about 45 minutes, and then we will see the gel under the transluminator. We are ready to load the gel now. The 30 minutes time for restriction digestion is over, and we have taken out the tubes from incubator. Now Aruj will be putting this gel in the gel chamber, where she will also put the buffer on it on the top, and then I will show you later how she deals with the digested samples before loading into the wells. So you are quite familiar by now about all this process of Agarose Gel Electrophoresis. She will pour the buffer, which is 1 XTA buffer, into these buffer chambers, both on the two sides of the gel. And there will be some of the buffer which will be overflowing on the top of the gel to complete the circuit between negative and positive electrodes. Yes, it is done. And she can remove the comb now. Okay. One will be loaded with 1 kb ladder. The second one will be loaded with the genomic DNA, and the third one will be loaded with the plasmid DNA, which is digested with PBU2. Genomic DNA is also digested with the same enzyme. We will run this gel at 60 volts for about 30 to 45 minutes, and then we will observe it under UV light. So Aruj, you are ready for loading. So what she is doing, that she will be adding now the gel loading dye to both of these tubes so that these samples can be easily loaded into the wells. She will be taking about 2 to 3 microliters of the gel loading dye for loading this all material onto the wells. So she has added the dye to one of the tubes, and she will be doing the same for the second tube as well. She's just mixing. Okay. So to the next tube, she will take some more dye. So after mixing the dye in the second tube, both the samples, they are ready. And now it's the time to load our wells with these samples. The first one will be 1 kb DNA ladder. We have been taking about 7 to 8 microliters from a premix which comes from the manufacturer. We directly load it onto the gel.
Microliters of sample.
15 microliter of the sample will be good for visualization. And 7 to 8 microliter of the 1 kb DNA ladder will be enough for seeing the ladder. So what are you loading?
Plasmid DNA first.
Okay, so she will be loading the plasmid DNA first. Okay, into one of the wells. Yeah, it's beautifully loaded. So the orientation now is changed. The first well has been loaded with the restriction digested plasmid, and the second well is going to be loaded with the genomic DNA, right?
जी.
Okay. So the second well is being loaded with the PBU2 digested chromosomal DNA. And now is the turn for 1 kb DNA ladder. So that's a very nice loading. You can see the dye inside the wells. It has not floated out. And now the marker is being loaded on the other side of the digested plasmid DNA. Okay, so we have 3 loadings done. So you can see it. The plasmid DNA is visible by a lower amount of dye. That is the 1 kb ladder, okay? The plasmid DNA is next to it, and then is the genomic DNA. So the 3 wells have been done on this gel, and the gel is now ready for running at 60 volts. Okay, now she's assembling the gel electrophoresis apparatus. The lid has been placed on the top, and she will be fixing these wires. There are 2 wires, one is black and the other is red, and they are fixed according to the color of the electrodes. The red goes to red and the black goes to black. So the voltage has already been set to 60 volts. Okay, she has turned on the power supply and it's going to now 60 volts. It's running at the moment, and we can see actually the bubbles which are coming out of the electrode at the black side, which is the 2 hydrogen molecules which are released as a result of splitting the water molecule into its ionic forms. Okay, and towards the other pole you can see the bubbling of I will try if you can see the bubbling of oxygen from the second electrode. Yes, you can see the bubbles along with the wire on the left hand side. So you can see the migration of the blue dye very slowly into the system, and this will run for about 30 to 45 minutes, and we will leave it like this till that time, and we will come back. Obviously we will not see the top as clear as we are seeing it now. The water vapors which are evaporating from the buffer, they will actually cover this transparent region, but I will try to see it after removing the lid again to look at the migration, that how much dye has migrated, which will give us the reflection of the migration of DNA. It's about 30 minutes that the gel is running now, and the dye is hardly visible, but it has gone about 2 or 3 centimeters away from the well. So we will stop the gel at this point, and we will open the lid to see how far the dye has gone the wells. Yeah, it's quite far from there. Yes, I can see the wells, and I can also see the dye migrated away from there. So we will take this gel now to the gel documentation system to observe the DNA in it. Okay, let's go. So we are back for visualizing the gel on the UV documentation system, and this system might seem very familiar to you now. Can I see it inside? So we have a transluminitor here, which is used actually for pressing the gel. So the gel goes there without gel caster. It is placed on the transluminitor this way. And now the door will be closed, and we will see after illuminating with UV that how it looks like. So you can see actually the orange fluorescence coming out of the gel. You can see on one side is the ladder, and in between is the plasmid, and on the right hand side is actually the genomic DNA digested with. It's not that clear as we can see it on the monitor. So if we can take the picture on this monitor, so you can see that, as discussed earlier, this plasmid was expected to give us two bands, one of 1.5 kilobase and the other one equivalent to 10 kilobase. If you look at the chromosomal DNA, we were expecting smear in this case, okay, and you can see the smear very clearly. So there is some RNA in there which is at the bottom of the lane, but if you look at the top, it is mostly the smear. It is due to the presence of a lot of PVU2 sites in the genomic DNA of wheat. Okay, so we will try to save this picture so that you can keep it in your records. And that's the end of today's experiment.